Right, there is the equation for respiration. Okay, so we've got glucose oxidized with oxygen to form CO2 and water. Now that reaction is very exothermic. Get a lot of heat out. But we don't, and by in cells, they don't want to waste all that energy, all that chemical energy in the glucose. You don't want to have it all coming out as just heat energy. You need to convert it into another form of chemical energy of ATP. And we're all pretty familiar with that. So ATP is, you can say it's a high energy compound. What that means is when it gets hydrolyzed by water to ADP, that's very exothermic, and you can use that energy to drive reactions in the cell. There's another high energy compound which we need to talk about, and you probably haven't come across it before, and this is called NADH. It's actually nicotinamide adenine dinucleotide. So it is a nucleotide, but its job is to all to do with energy. So NADH, right, that is the reduced form of it. When it's oxidized, it's written usually as NAD+, it's got positive charge on it. And the oxidized form does not much chemical energy in there. So that's low chemical energy. But the reduced form, it's got a lot of chemical energy. And we'll see later in the mitochondria, the high chemical energy of NADH is used to make ATP. So NADH is like an intermediate high energy molecule, which we need to make. And we'll see at various stages in respiration, we make this reduced form of it, the NADH, and that's a high energy, and that can later, we can use that the cell uses that to make ATP. So just in case you haven't come across NADH before. Okay, so let's see how this works then. Right, now we can we're going to split this into, in this video, into 3 stages. And the first one of those is called glycolysis. Okay, so this is like the first one here is glycolysis. Let's see what happens here. Right, what I've drawn there this is glucose. Right, you probably recognize, so there's 6 carbons in it, C6H12O6. And here, well, this is a fructose molecule, but now it's got 2 phosphate groups. It's got 2 inorganic phosphates, which are like that, are written as P. Of course, that is really PO4 3 minus. Okay, so that's the inorganic phosphate group. Now, fructose is an isomer of glucose, so all that's happened there, you rearranged the carbon atoms. But to put these 2 phosphate groups on here, that one and that one, you need ATP. So the whole idea of respiration is you're going to make some ATP, but before we start, you have to put some ATP in. You're going to need, for each one of those phosphates stuck onto the fructose, you're going to need an ATP, and that will become an ADP. So that's an initial sort of input that you've got to make. Okay, so now we come to these diagrams here. Okay, now those blue circles, each one's referencing a carbon atom. So the 6 carbon compound of fructose, so that's C6H12O6, has now been broken into 2 3 carbon units. There. And each one of those has got a phosphate group attached to it. Okay, now that 3 carbon unit is oxidized by NAD+. And in the process of NAD+ oxidizing that, you get NADH made. So in this stage here, you end up with 2 molecules of NADH, which is 2 high energy intermediates. And also, what happens in this stage also is you transfer that's an inorganic phosphate there, so it's not from ATP, and you stick that onto the ends of the molecule. So you've now got 2 phosphate groups per on each of those 3 carbon molecules. In the next step, what happens is those phosphate groups are transferred onto ADP. So you end up making 2 molecules of ATP. So if you think what's happened there, you started off with inorganic phosphate, and by binding it to this 3 carbon unit, you've now managed to transfer it onto an ATP. So that's good because you've getting some high energy intermediate. Now you can see that we've made 2 ATP molecules, so we've broken even. We had to input 2 ATP molecules at the start per glucose, but we've got 2 back now, so that's even. So anything else now is going to be profit. Okay, so the next step then is, those very similar, these 2 phosphate groups, each of those is transferred to an ADP molecule to make 2 more ATPs. And this final molecule we end up with is called pyruvate, and it has a formula called pyruvic acid, C3H4O3. I won't write down what the formula is, but we don't really need to know. Right, so let's just see what's that. Now that whole process of going from glucose to pyruvate, so right here, here's glucose, and we've turned it into 2 molecules of pyruvate. That whole process of doing that is called glycolysis. Right, let's have a look at the oxygens and hydrogens in there. So if you can see here, we've got 6 oxygens in glucose and 2 lots of 3, you've got 6 in pyruvate. So the amount of oxygen hasn't really changed. But we have a look at the amount of hydrogen. You've got 12 hydrogens in glucose, but here we've only got 2 lots of 4, so that's 8. Now when you lose hydrogen, that is the same thing as oxidation. So the glucose has been partially oxidized there. It hasn't been oxidized all the way to CO2 and water, but it's been partly oxidized. And whenever you carry out oxidation reactions, if you think of burning things, they're nearly always exothermic. So glucose turning into pyruvate is an exothermic reaction, but we haven't wasted the energy as heat. What we've managed to do is get that energy stored in these high energy compounds, ATP, and which we're familiar with, and this other one, the reduced form of NAD plus, NADH, which that can go into the mitochondria and be used to make ATP. So that's the end of glycolysis. Now the second phase is to do is called the link reaction. Because the reason why it's called the link reaction is because it links glycolysis to the third step, which is called the citric acid cycle. We'll do in a minute. Right. So let's see what we've got here. We've got pyruvate, a 3 carbon substance, and here this stuff is called coenzyme A. Now it's a fairly reasonably complicated molecule. It's not a protein. It's a vitamin. It's actually one of the B vitamins. You need it in your diet, and it plays an essential role in carrying around. Well, you can see it actually carries this 2 carbon unit here, and it's absolutely essential in respiration there. So coenzyme A reacts with pyruvate, and you lose one of those. Can you see you've gone from 3 carbons to 2 carbons here? So you lose one as carbon dioxide. So this is the first sign we've had so far of carbon dioxide being produced. We know the overall reaction, it goes to carbon dioxide and water. Well, here we are, we've got our first carbon dioxide. Obviously, we've still got these 2 carbons. They've got to be turned to carbon dioxide at some point, but that will happen later on. We also see what happens when pyruvate is converted to coenzyme A. If you look here, we make NADH. So you can, what actually happens is, so that is, well, the NAD gets reduced. So that means the pyruvate is being oxidized actually. But the important thing is we make another high energy molecule here, NADH. Right. Right, so just to show you that that is oxidation, if you look at the number of oxygens here, we have got 3 oxygens on this side of the molecule in pyruvate. By the time you get to that side, you have got 1 there and 2 there, so that's the same. But the number of hydrogens has gone down, so that is oxidation. Yeah, because look, you've lost hydrogen. Got 4 hydrogens there, you've only got 3 there. So it is oxidation. And the energy of that oxidation is the energy is trapped, not wasted as heat, but trapped in NADH. Now, this coenzyme A, this is actually this is called acetyl-CoA, this stuff here. Right, this acetyl-CoA molecule has got to feed into the citric acid cycle. And we've got, this is the citric acid cycle here. So if you look at it, it's a circle, so we've got the whole thing together. Right, so how does it work then? Right, before we look at it in detail, what we can see is we've got a 2-carbon unit going in there, and it goes round, and by the time it gets here, you've only got 4 left in it, so you've lost 2 carbons, and they've come off here. Look, 1 carbon dioxide here, and the other carbon dioxide here. So now all of the carbons in the glucose have, by that time, have all been converted into carbon dioxide. We lost 1 in the link reaction per pyruvate. We're losing 1 there. We're losing 1 there. Of course, this happens twice for each glucose molecule, so that's a total of 6 CO2 molecules all gone. So that's where the CO2 goes. And let's see what else happens. Right, we start off with this stuff, oxaloacetate. You don't need to know the name, but it's got 4 carbons in that molecule. Right, acetyl-CoA, here we go. That sticks on those 2 carbons to make a 6 carbon there. We've got 6 carbons in citrate. And what's going to come off here is the free coenzyme A. Right, then citrate goes to this stuff, alpha-ketoglutarate, and that's, you can see you lose 1 of the carbons as carbon dioxide, and also you make an NADH molecule, another high energy molecule that you've made there. Once you go to alpha-ketoglutarate, then it goes to this succinyl-CoA. So this is only 4 carbons. So you have lost the carbon dioxide molecule, and again this is an oxidation reaction because you end up making another molecule of NADH. The NAD plus is the oxidized form, and that oxidizes the alpha-ketoglutarate to the succinyl-CoA, and it becomes reduced itself in the process, going to NADH. Right, now this is where you get a bonus molecule of ATP, because this succinyl-CoA, well, it's the same coenzyme A, right, so you must have a CoA coming in here. Free CoA comes in here. The CoA is this time not stuck to a 3-carbon unit, but stuck to this succinyl, this 4-carbon unit here. And in the next step, this one here, the CoA gets split off. So there your CoA goes out. And that's quite an exothermic reaction. And instead of wasting that energy as heat, it's trapped not into an ATP molecule, but a GTP. So that's guanosine triphosphate. So it's one of the bases in DNA, as is adenine, and so guanine is a base. It's a very similar molecule to ATP. It's a high energy compound, just like ATP. So at that stage, we've now got 4. We've got 4 carbon atoms. We don't need to lose any more carbons, but it undergoes some further oxidation reactions. I'll talk about this one, FAD, in a minute. But you create another NADH molecule there, and here you make this stuff FAD, which is very similar to NAD. It's that's the reduced form, and that's got a lot of energy in it. And FAD without the plus this time, that's the oxidized form, and that is not got much energy in it. So, and FAD, just like NADH can be used to make ATP in the mitochondria, this stuff, FADH2, that is used to make ATP in the mitochondria. Okay. Incidentally, I should say where are these reactions occurring. So the citric acid reactions are all in the mitochondria. And if I just scroll up here, right, there we go. Glycolysis, this bit where you end up with pyruvate, so this all occurs in the cytoplasm outside the mitochondria. The link reaction, that occurs, so the pyruvate has to go into the mitochondria, gets transported into the mitochondria by a special membrane carrier, and the link reaction then occurs in the mitochondria, and the citric acid cycle, that also occurs in the mitochondria. Okay. Now, let's just see what's happened overall in the citric acid cycle. So overall, we started off with acetyl-CoA, which is there, and that has the formula CoA. Don't worry what that is, but it's C2H3O, and we turn that into free coenzyme A here and 2 carbon dioxide molecules. So if we look what's happening here, well, we've gone from having 1 oxygen here, and we've got 4 oxygens. So there's definitely been a lot of oxidation going on there. Also, we have got 3 hydrogens on the left, and we haven't got any on the right. So they've actually gone to water, but we won't worry about that. So that means this is definitely an oxidation reaction. It's pretty exothermic, that reaction. If you carried it out, you get a lot of heat out, but you don't waste the energy as heat. You trap it in these high energy molecules, the reduced NADH and the reduced FADH2. And the next part of the of respiration, which we'll cover in the next video, we'll explain how they give you ATP. But we'll just have a look at the yield there. What have we got so far? Glycolysis, we got this is per glucose molecule. We've got 2 ATPs and 2 NADHs. The link reaction, well, we got 1 NAD per pyruvate, but of course you get 2 pyruvates from 1 glucose, so you get 2 NADs there. In the citric acid cycle, you get 3 NADHs and an FADH2 and a GTP. That's per acetyl-CoA, but you get, of course, 2 of those from each glucose. You've got to remember that. So that adds a total. So far, we've got 4 ATPs and 10 NADHs and 2 FADH2s. And what's going to happen with these is they are going to be used in the mitochondria to generate ATP. Now, if I just show you here, NADH, I said this is high energy, and it is reduced. And this is NAD+. This is not hasn't got so much chemical energy, and it is oxidized. Right, in the mitochondria, it's very complicated, but it gets oxidized by oxygen, and you end up with H2O. So this is where oxygen comes into respiration. We need it here to reoxidize the NADH up to NAD+. Now that is a very exothermic reaction, but of course it is not wasted as heat. It is used to make ATP from ADP, so you trap all that energy in the ATP molecules.